what enzyme engineering is, system biology is, synthetic biology is. Now, you can implement those to tweak your hosts or even tweak your product or type of product that you are expecting after the fermentation process. But the question would be, what sort of reactor will you choose? And what are the different types of reactors that are available, why we actually look into bioreactors in one way or other, and what would be the design principles that are involved when you are actually designing a bioreactor for a specific purpose. Now, before I start the lecture for today, I did receive a question with respect to the case study 2 that we were discussing yesterday. Do you all have any questions with respect to what you are going to do in your case study 1 or case study 2? What I have provided you with respect to the case studies was the product. What you have to do is go backward, develop the whole process. Okay? Now, the product that I have stated might not be the final product. It might be just the product that would have received from the industry, but it cannot be consumed. Let's say monoline cannot be consumed if it is not heat stable, right? Enzymes are also not heat stable. So, the alpha amylase that you will produce, how do you want to sell it? If you want to sell it, do you want to encapsulate it? Do you don't want to encapsulate it? What would be the optimum way of producing the final product has to be decided by you, not me. Okay? Let's start the lecture for today. So, we are going to start our conversation with bioreactor design and analysis, but before we get into the design and analysis part, we will talk a bit more about the bioreactors. We have covered majority part of the bioreactors, how they are implemented or how they are used with respect to your cellular agriculture and precision fermentation, but we will I'll say like we'll go through it again. So, bioreactors, as you can see from the picture here, those are the huge ones, and all those pipes that you see here, those are the controls or any of the inlets that are providing the substrate inside the bioreactor. So, it is a pretty complex unit. When you look at it, it is a very complex unit. When you operate it, it actually looks very complex, but it is a very simple system. In principle, it is a very simple system. What it does is very simple, but the way it has to be operated is very complex, and that is the reason why you have to have the engineering design principles involved here, and we need to understand how to design a bioreactor. Now, we are using enzymes, plants and animal cells, or we are even using microbes as our hosts. Enzymes, if you are just providing a substrate, you put in the enzymes, you can get the substrate converted into the product directly. Plant and animal cells, we have already discussed multiple times, they can be used as a host to produce your product of interest. Microbes are widely used as a source, as a host to produce your product of interest. So, then there are different types of products that we do develop. So, there will be the bioconversion products, where you have the substrate being converted into product. Biotransformation, where one substrate has been transformed into a product, and the recombinant products is what we have discussed as part of the precision fermentation, where we are developing the product using recombinant DNA technology. So, we have genetically modified our host to produce the product of interest. So, a bioreactor by definition is a device in which a substrate of low value is utilized by living cells or enzymes to generate products of higher value. Now, substrate of low value, if you are using pure glucose or pure fructose or galactose, they are not cheap. They are abundant, but they are not cheap. When we do talk about low value substrates, we are thinking about the feedstock. So, let us say if we are able to utilize the agricultural waste. So, that is the reason why I give this particular definition there, but majority of the times the final product that you are developing, that is of super high value. So, that is why the substrate cost does not matter much, even if you are using galactose, sucrose, fructose to produce your product. If the final product has a way much more higher market value, your substrate cost will not be of any concern, correct? That is the same case that you will have in your case study 1. If the monolin is able to capture the whole market or the whole food industry market with respect to the sweetener part, that particular product is going to have a pretty high value, a market value. So, your capital cost involved in terms of choosing the substrate might not be of concern to you. Okay? So, be wise. When you are doing the techno-economic analysis, you would be able to figure it out, what should be your input, what should be your output, and how you are going to do that. Now, the bioreactors have been used to produce different types of products, some of those that I have mentioned here. So, vitamin B12, those are produced through genetically enhanced microbes that develop it through one-step fermentation process using vegetable oil as a feedstock and sugar as a nutrient. We have genetically enhanced microbes producing baking enzymes used to enhance rising, strengthen dough, and prolong freshness. We have already discussed how the enzymes will basically provide the sensory aspect to our products, right? Microbes and fungi are genetically enhanced to produce enzymes that are added to as brightening and cleaning agent. We did discuss about that. The protease enzymes and the lipase enzymes. The lipases are mostly used to remove grease. You would have heard about not natural, but biocompostable detergents. They are not the regular detergent, liquid detergents, but they are like compatible, more environmentally friendly. They actually have more of these enzymes, genetically enhanced enzymes or genetically produced enzymes. Now, bacillus microbes ferment corn sugar to make lactic acid, which is heated to create biodegradable polymers for woven fabrics, and these are the ones which are used to make diapers for kids, for babies. Now, this example, this particular slide shows different examples of fermentation system, like how fermentation has been used, but majority of the examples that I provided are more inclined towards a recombinant DNA technology, right? Always using a genetically modified organism to produce a product, because currently that's the norm. The regular fermentation, we are using Saccharomyces cerevisiae to convert barley into alcohol, right? Those will be the regular fermentation, the traditional fermentation. So, traditional fermentation is still going on. They still use the same type of bioreactors. Even in there, you have to choose whether you're going to go for a specific bioreactor, which might be a batch reactor or a fed batch or a continuous stir tank reactor or even a plug flow reactor. So, there are different types of reactors that you can use. Now, what will you use for majority of your precision fermentation processes? That is what my interest was, and that's the reason why we have these examples. Okay. So, biochemical synthesis versus chemical synthesis. Why biochemical synthesis is better is because it is mild reaction conditions. You are not using very harsh reaction conditions. If you remember from the slides of yesterday where we are producing the PTA, at that time we were looking at cobalt, manganese being used. Those are harsh chemicals which are basically being used. It has a very high regio and stereo selectivity, because if you are using enzymes, you have the capability of this. It's sustainable if you are using a sustainable feedstock. It has a better product yield. We did compare, if you remember, we did compare the PET with the conventional PET. Non-toxic and undesirable waste as byproducts. So, they are non-toxic in nature, and the byproducts which are produced are not toxic either, and they can be used further for other purposes if need be, and it is more environment friendly. So, the GHG emissions and all those things are reduced because you are changing the feedstock. By using a cheaper feedstock or a waste as a feedstock, you can reduce the overall GHG, and you can make it a very carbon neutral system. You know what carbon neutrality is? So, let us say you have plants. Now, you use the plants to make fiber, You use that fiber for making clothes. Now, once the clothes, like the production of the clothes, the CO2 which is generated. So, let's say this is the industry which is producing the clothes, they are generating the CO2. Now, this CO2 is again used by the plant to make, to grow, right. So, basically it becomes a carbon neutral cycle. So, that is what I am talking about. So, it is more environment friendly. High regio and stereo selectivity. If you are using enzymes, enzymes have the lock and key functionality, right. So, depending upon the active sites, it might, and the substrate that you are going to use, it will be very selective to that particular substrate. So, if you are using multiple substrate, but you only want one substrate to be used, you can actually use that. So, that's what it is trying to say. So, you have the regional and the stereo selectivity.
Thank you, sir.
Now, this is an example of a typical bioprocess that you can see. So, you have your media and nutrients which are coming in through ultrafiltration into a bioreactor. You have water which is being added to the bioreactor through reverse osmosis. Air is being passed in to provide the oxygen to the bioreactor. And then you have, once the cells have grown to a specific concentration, they are moved into membrane filtration. Now, after the membrane filtration, some of that cell go back as cell recycle, or you can harvest the rest of the cells. Some you put back, some you reharvest, and whatever product you have had after the membrane filtration, you can do the downstream process, so the downstream processing. So, this is your downstream processing. Now, this is where the maximum cost is. Now, just a quick question, why do we do a cell recycle? Any ideas?
Sir, when we operate at high dilution rate, we do not want our cell mass, biomass to decrease in a bioreactor.
Okay. You have gone way much more in advance. We have not reached the dilution rate yet in our discussion, but to a certain extent, yes, but more of to maintain the same culture. You don't want to change the batch of your culture, right? You want to maintain the consistency. So, what is the best example of cell recycle in your day-to-day life? Yeah, yogurt and curd, right, at home. So, you keep the culture. After you have finished the yogurt, you keep a little bit of it so that you can reuse it. So, that maintains the consistency. Cell recycle allows you to maintain that consistency. Okay? Oh, I could have asked another question. The media and the nutrients that are being added into the bioreactor, they go through ultrafiltration. Why? You need them to be clean, but you also need them to be sterile. Anything that is added into the bioreactor has to be sterile except the cells. Okay? All the nutrients and the media that is added into the bioreactor at all times has to be sterile. It cannot have any contamination, because the moment you have a contamination, you are going to change the whole profile of your bioreactor inside, right? Now, this is a typical bioprocess. I am not saying that this is a bioreactor design that we are following, but what type of bioreactor will this be? It has an input and it has an output. If it was a continuous input and output, it becomes a CSTR, a continuous stirred tank reactor, right? If it has only an input, it becomes a fed batch. If it has no input, no output, it becomes batch, right? Most of you have the background in biotechnology. You would have taken a course in bioprocess, so I know that you are aware of it. For those who don't, there are three types: batch, fed batch, continuous. So, design of a bioprocess depends on a couple of things. The most important would be how fast will the process take place. So, the reaction kinetics. What changes can be expected to occur, like what would be the metabolic state, what would be the cell physiology that would be reached, and how will be the system be operated and controlled to provide the maximum yield and productivity. So, what sort of design of reactor we are going to use and what sort of instrumentation and control we are going to have. So, what would be the temperature profile, what would be the pH profile, how much oxygen is going to be purged, what would be the speed of the impeller within the bioreactor, all that has to be controlled. That comes under the topic of process control and instrumentation design. If you are looking into chemical engineering, that would be process control and instrumentation design. So, the other aspect of the design of a bioprocess would be how will be the products be separated or recovered with maximum purity. So, the downstream processing. Now, we have discussed to a certain extent in detail about downstream processing. We did talk a little bit about the temperature and the pH yesterday when we were discussing about the enzymes and how they might impact the structure of the enzymes. So, if let's say I am assuming that you are aware what a cell culture looks like. Okay? So, if you have a flask, have you ever seen a flask with a cell in it? In the microbiology, if you have taken a course in microbiology, you would have seen that, right? Now, if I heat it, what will happen to the cells? They will die, right? So, temperature, so the moment, let's say if the cell cultures were at 25 degrees Celsius, that's the normal room temperature right now, okay? So, 25 degrees Celsius, which is a normal room temperature. If I slowly increase this temperature all the way to 70 degrees Celsius, will they start dying the moment I start increasing the temperature, or will they die after we have reached a specific threshold temperature? So, once we have reached a threshold temperature, right. So, temperature actually sometimes will promote cell growth. Eventually, once it has reached a threshold, it will start dying. So, you will see that the cell will start growing, and then, and this would be the temperature at which, or T max, that you can actually have. Now, the reaction kinetics, like how fast will the process take place, it is very, very important. If you are looking at from the chemical engineering perspective, the chemical reactors, you are using chemicals as your substrate, and the product that you get is also a chemical. You are providing a catalyst, you have to control the temperature and the pressure, everything. Now, some of the reactions are super fast, some of the reactions take time, right, and that's the reason why we add catalyst, so that we can increase the speed of the reaction. Then some of the reactions are exothermic, some of the reactions are endothermic, right. When they are exothermic, what does that mean? Thank you for the unity. I would prefer if one person can answer that.
Energy will be needed, endothermic.
So, when you are releasing the energy into the system, that's exothermic. So, that means you are increasing the temperature of the system. Am I correct? Endothermic, you are absorbing the energy. Does that mean that I am reducing the temperature of the system? Yeah.
Exo.
Yes or no?
Exo will release, endo will take.
So, if I'm doing an endothermic reaction, does the temperature of the system decreases? Thermodynamics 101. Tell me. What will happen to the entropy of the system in an endothermic reaction? It will reduce, right? The temperature might not. There are different exceptional cases, but temperature might not. Okay. So, your thermodynamics is now involved down here. You have to have a background in thermodynamics if you want to do a bioreactor design. So, engineering background is very much required. Now, what changes can be expected to occur when we are looking at the metabolic state, the cell physiology. So, if we are aware of how which metabolic pathway is going to be followed by our cells, we would be able to provide them with the right nutrients which would be required for that metabolic pathway to get you the product of interest. Okay? So, understanding that will also help. Understanding what is the growth rate, or how quickly our cells will double, what would be the physiology, what would be the concentration of our cells later on, will also help in designing the bioreactor. Understanding that the thing that I'm trying to state here is that whether I'm going to choose an E. coli or whether I'm going to choose an yeast, that depends on the product that I want to grow, but it also depends on how well they grow, right? So understanding that is very, very important when you're designing a bioprocess. Now, what is an efficient bioprocess? Whenever we talk about efficiency, efficiency has to be defined in terms of time. Efficiency can be defined in terms of money. If it is able to make you more money, it's an efficient process. If it is able to save you money, it is an efficient process. If it takes less time, makes you more product, it's an efficient process. Other than that, it also depends on the production of organism. It depends on the optimal conditions for the desired product. If you're using the optimal condition for a desired product, you're going to have a very efficient bioprocess. The product value will define your efficiency of the bioprocess, and the scale of production. If you're able to produce in mass, or a grand scale or a large scale, you would be able to make more money out of it, and your process would be an efficient process. Like setting up a startup, you should be able to make some money at the very end. So this includes, so when we are trying to do the efficiency calculation, we have to look into the size of the reactor, the type of the reactor, and the method of operation that we are going to use, which will give you the best for a given conversion. So if you're converting substrate A into product B, you should make the right choice in terms of choosing the right reactor, the right size of the reactor, right? A single person does not need an airplane. A lot of people will require an airplane, let's say. If I'm a billionaire, I will definitely need an airplane, but I'm just saying in terms of, like, as a single person, I might be happy with a bicycle to reach from point A to point B. So depending upon your need, you have to make the right choice in terms of choosing the right reactor, the type of the reactor, the size of the reactor, because all of that will define how energy efficient your process would be. If you're choosing a bigger reactor for a smaller reaction or for a smaller product size, your efficiency is less. Okay. Now, the bioreactor design, there are a couple of other things that are involved in it. So those would be your control and the positively influence the biological reactions, and you have to prevent contamination. The capital investment and the operating cost, we already discussed that. So during fermentation, maintenance of monoseptic condition is very, very important. So anything and everything that is added inside is sterile. So the nutrients that are going in are sterile. So you're only growing one microbe inside the microbe that you have put in, the culture that you have put in is the only thing that is growing inside the reactor. You're not adding any contaminant to it. You're not adding any other microbial colonies to it or any other microbial population to it. So optimal mixing with low and uniform shear rate. Now, why do we need optimal mixing in terms of a bioreactor, and why do we need a uniform shear rate? Optimal mixing will help to homogeneously distribute the nutrient and oxygen if you are sparging oxygen. Uniform shear rate, if you are able to identify the optimum shear rate or optimal movement of the sparger or the rotational speed of the sparger, the mixing would be ideal. You are not going to break your cells. If you make your your impeller. If you make your impeller to run fast, you might break your cells. That will lead to cell lysis. Then comes the aeration, surface or direct sparging or indirect. So depending upon what will provide you the best and optimum O2 distribution within the bioreactor, if it is an aerobic, if it requires aerobic conditions, you need to identify the best way of providing that aeration into the bioreactor. So maintenance of adequate heat and mass transfer and the flow conditions. So let's say for a fed batch, we have to have an input. Let's say for CSTR, you have to have an input and an output. It has to be maintained in such a way. You cannot have a lower output, higher input, because then your reactor will overflow, correct? So, you have to maintain that optimal condition, the balance, so that your cell, the cultural growth or the cellular growth is maintained within the reactor at all time. The products and the byproducts removal is also there. So, to maintain the optimal conditions within. Now, bioreactor, for that we have to look at, like, it provides a controllable. So, my mistake. So, what does bioreactor do? So, bioreactor provides you a controllable environment, enabling the biochemical and the biomechanical requirements to create a desirable product. So, you are being provided with both the parameters to grow your product to the best of its capabilities. It enables close monitoring and control of the reaction parameters like internal and external mass transfer, heat transfer, fluid velocity, and shear stress. So, one unit operation can provide you all of this control, and then you can grow your cell culture within that unit operation. This is a diagram of a typical bioreactor. The previous one that I had shown, that was a diagram for a typical bioprocess. This is a bioreactor. So, what are the parts and what are the most important parts for us would be your agitation system, that means your impellers. The design of your impeller will also be a parameter at one point of time. The sensors, the sensor probes would be for your pH and the temperature. So, you are looking at your pH, you are looking at your temperature. What else can I use the sensors for? Dissolved oxygen, right? DO. What else? The level? Let's say it's a batch reactor, so the level is maintained. So, you're looking at the dissolved oxygen, you're looking at the pH, you're looking at the temperature, right? These are the major sensors. What else can be used? Or what else can I sense inside a bioreactor?
Conductivity. Conductivity.
Conductivity? Why? And in what sort of application will you use conductivity sensors?
Majorly conductivity measure, like for insulin production, they measure the conductivity to consider how much salt concentration, how much is available in, or how much.
If you change your salt concentration, that will change your pH also.
Yes, but majorly in industrial level, conductivity also one of the parameters that can they.
They do use conductivity meters, yes. The conductivity sensors are there. I personally think if they have been used when you are working with microwave fuel cells, mostly.
Cell counting sensors, NIR probes.
Have you ever seen
I have seen them.
a bioreactor having that?
Yeah, I have seen that.
So, when they are doing the NIR probes, you said?
NIR probes. I don't know the full form, but they are used to measure the cell, like what is the cell count at particular time.
Okay. I haven't had that experience yet. So, thank you. So, there are a lot of many sensors now that we can use inside a bioreactor. So, you have your reactor tank, that's a typical design of a bioreactor. You have your feeding pump, which is basically used to push in the nutrients inside. You have your spargers, which is going to provide you the aeration. You have your agitation system to maintain the homogeneous distribution of the nutrients and the oxygen. You have the thermal jacket. Now, this thermal jacket is to maintain the temperature within. Okay? So, this is a typical diagram. So, parts would be agitator, baffle. Now, baffles are sometimes here in the corners of the bioreactors. In this particular diagram, it was not there. Now, why do we add those baffles? To remove the vortex, right? So, because when you are running your sparger, sorry, when you're running your stirrer or your impeller, you generate the vortex. To break that vortex, you're going to use the baffles. So you have your sparger, the cooling and heating jacket to control the temperature. You have your control systems. Typical ones would be temperature, pH, and dissolved oxygen. And thank you for the cell count part. I will add it. Now let's get into the classifications of bioreactors. Now bioreactors can be classified based on the presence of or absence of oxygen. Before we start discussing that, can you tell me the two types of bioreactors that we have actually discussed once on the day one? I asked you a question about if you have potato peels, or let's say you have orange peel, what sort? So either it would be a submerged bioreactor or solid state, right? So that's also another classification. So whether you're going to have everything submerged inside or not. Then you have your in the presence of oxygen or in the absence of oxygen. So anaerobic fermentation, so no aeration is required. Initial preparation of inoculum may require aeration because you want to grow the cells. The gas released during the fermentation is sufficient to provide the mixing. So when you are in anaerobic production, you will have some gas generation within the reactor, inside the reactor as the cells are growing, and that gas bubble is going to move up. As it is going to move up, it is going to make the mixing of the nutrients with the cell culture. The product recovery may also require anaerobic conditions. Majority of the times, if I remember correctly, in the lab we provide anaerobic condition by purging with nitrogen. For aerobic fermentation, you have non-stirred aerated reactors, which would be airlift bubble column reactors, in which you are basically, so there is no impeller, so it's mostly being mixed with the aeration. Stirred and aerated reactors, where you have the stirred tank reactors, so CSTR would be a part of that. Some of the batch reactors would be part of that. Fed batch are also part of that. So on the right hand side, you can see an example of airlift bioreactors, where you have the draft tube internal loop configuration, a split cylinder device, and then you also have an external loop system. So you will see majority of these things are moving. A very similar design for this would be, if you have learned dehydration of food products, there is a design of one of the dryer like that. What is the name of the dryer? Where your product is fluidized. We call it fluidized bed dryer, right? So it's because you want to make sure that your air is uniformly distributed, your product will dry faster in that case, right? It's very simple, similar concept applied here, or this concept was applied there. Now based on the modes of operation, now this is what we have been discussing. I have actually stated these names multiple times. So it can be a batch reactor, it can be a fed batch, or a continuous stirred tank reactor. Now when it is a batch reactor, no input, no output, so it's like a pressure cooker. You put everything inside, close it, let it run. Once it's done, gives you the signal, take your product out. Okay? In a fed batch, we can add the nutrients. There is no output, but we keep on adding the nutrients. So the intent of a fed batch would be to reach the maximum cell mass, right? We keep on adding, the cell will keep on growing. The cells will grow continuously till the time all the substrate that is present inside the bioreactor is consumed. If you keep on adding the substrate, the cell mass will keep on growing. So the intent of the fed batch reactor can be to have the maximum cell mass. So now from your own understanding and experience that we have had through conversation in the past couple of days, where will you use a fed batch? If the product is intracellular, right, developed intracellularly, you want to have the maximum biomass that you can take out, crush and take your product out. That can be one application. Where else will you use it? Will I like to use it in let's say if the product was extracellular and the cell can continuously produce it? In that case, I can go for a continuous stirred tank reactor, right? So, depending upon the type of product that you are going to develop, you can choose the right type of reactor, right? So, in the continuous stirred tank reactor, you have your S0 and X0. Your S0 is basically your substrate concentration, your substrate at time T0, that is initial substrate concentration, or the substrate that is coming inside. Then you have your X0, that is your cell biomass concentration at time t is equal to 0, your initial cell biomass concentration. X, S and P are your X would be your cell at the end of what is coming out, S would be what the substrate that is coming out, and P is the product. Now, in a continuous stirred tank reactor, if you see the output, you have your cell, you have your substrate and you have your product, correct? So, you still have your substrate. So, not 100% of the substrate has been used. The cell that is coming out is going to reduce the cell count inside. So, you can do a recycle in that case. So, that is the reason in CSTR we generally have a recycle. So, what are the design considerations for a bioreactor? The most important for any startup in the world, capital. Similarly, if you want to set up a bioreactor facility or any fermentation facility, the very first thing that will come into picture would be your initial capital expenditure. Now, out of all these three reactors, which one do you think is the cheapest? Batch, because of ease of operation. Or you can buy as big as you want, you can put as much of your substrate into that, let it run, and once it is done, open it, take your product out, right? So, substrate, if you see, that is why I am saying, like, if let us say I come to you and say that, okay, I need this particular product, your choice of reactor will depend on many things. One of the things is how much money I am going to give you as your initial capital, right? So, your initial capital expenditure will define the type of reactor that you are going to choose. The ease of operation would be the second most important parameter. Less downtime. Now, what is your downtime? What is downtime? So, you have a reactor, all the reactions are done. So, let us say it took, so you started it now, it took 3 or 4 hours for the whole process to end. After 4 hours, you removed, you open it, you remove your mass, cell mass, you clean it. It takes you 2 hours to make it ready for the next batch. The downtime is 2 hours, right? Run time 4 hours. Okay? So, we will be using these terms like downtime, the T, and the run time, or the batch time would be your 4 hours, downtime would be your 2 hours. So, TB would define your batch time, TD would be defining your downtime. So, using these parameters, you will be able to understand the efficiency of your process that you are working with, or even the productivity. You can define productivity not just on the terms of how much cell mass you can produce or how much product you can produce. You are also going to look at the total. Like if you're running your reactor every day for 365 days of the year with a specific downtime in between, your productivity will depend on how many times you have ran it, how much of the downtime you have had in between, because that will define your total time. So, total time is batch time plus TD, simple. So, how if I tell you like you are able to run 2 batches a day, if you are running your system for 365 days, how many batches did you run? 365 into 2, and that will give you the total mass you would have accumulated, right, over the year, or what your yield had been over the year. Maintenance, ease of maintenance, not just maintenance, ease of maintenance is also very, very important parameter when you are trying to choose a bioreactor or even design a bioreactor. Skill requirement, there was a question I remember. So, skill level that is required to run a bioreactor depending upon the choice of bioreactor will differ. If you add too many complexities, you need somebody who is highly skilled, right? If you have less complexities, you can run it without any skill. Give me one very good example of a batch reactor, not a continuous stirred tank reactor or let's say any of the reactors which have been operated by least skilled people, a product that was developed by least skilled people. They don't have to have a degree in engineering. Come on, you do that every day at home, your mom does that, yogurt, curd. Another in North America it would be moonshine. Have you heard about moonshine? Illegal liquor,
Okay.
which you can do brewing in a jungle. You don't need a highly skilled engineer to be in the jungle, hiding from the police, brewing your fermentable alcohol. That's your moonshine. In North, at one point of time, it used to be illegal to actually brew moonshine, and people were still doing it. So that's a very good example. You don't need to have skill. You need to understand how the process works, right? Skill is if you can operate it. Technical skill is a different thing. So when you start adding complexities where you have to estimate how much of dissolved oxygen is there, are you able to maintain it, you have to do some calculations. Yes, technical skill is required at that time. That might change the type of reactor or the choice of reactor for you. If your choice is to make sure that a specific skilled level is utilized or employed for a process. The other would be your control systems, the type of controls that you would like to put. If you only require a basic control, that means temperature, what is happening inside, or you need your temperature, you need your pH, you need your dissolved oxygen, you need to know your cell concentration, you need to add all of those controls to design the bioreactor. Then you have your quality or the culture. The design consideration will require the quality of culture. Now, quality of culture matters a lot. What do I mean by the culture? So, if you remember when we were discussing about the process to reach the reactor stage, we have to grow the cells in the lab. So, we use the flasks, we grow in them in that. But then we are what are we trying to do? We are trying to grow the culture or the cells in the nutrient rich environment, right? So that their metabolic machinery within the cell has aligned to that nutrient, but that nutrient has to be similar to the nutrients that they are going to get in the reactor. Might not be in the same concentration, but they will be getting in the reactor. So that small scale, pilot scale, large scale, that's how it goes. So you have to have that chain. So you develop the culture, you build the culture, then you pass on the culture inside. Correct? Now that machinery, the metabolic machinery which is within the cell, that will change if you change the carbon sources. So let's say if I have given a carbon source 1 to the cell culture in the lab, and in the fermenter I am giving it a carbon source too, what will happen? Will my cells grow? Yes or no? If that carbon source can be used by the cells, it might grow. If that carbon source is not to be used by my cells, they will not grow, they will die. The quality of culture will also make a lot of impact, because if you have an older culture, again an example would be your curd and yogurts at home. If you use an older culture, you might not get a proper set curd. If you are using a new culture, very new culture, where you don't have enough microbes, you might not get a proper set curd. But you have to have the appropriate optimum culture to grow in the fermenter system, right, or in the fermentation process. When do you reach it? How do you reach it? It has to be done outside the fermentation process. Okay? Now, the classifications of bioreactors, you have your solid state bioreactors, the cultivations in the absence of free water, of pretreatment of the substrate, and difficult to control process parameters. Now, solid state bioreactors, the cultivation is in the absence of free water. Pretreatment of substrate and is required, and difficult to control process parameters are involved in this. It's not as simple as regular submerged bioreactors like a batch process or a fed batch or a controlled stirred tank reactor. Application of these are mostly for antibiotics, food additives, biocontrol agents, and bioremediation. In my research group, we have used it for food additives, specifically to get natural pigments. Natural pigments would be your natural colors from the food waste products. So, we were using onion peels, we have used carrot. In North America, you get here you have your carrots with the top, then you have your leaves, right? Excuse my drawing. When you go to the market, you can buy the whole carrot here. There, they will cut this part and this part, and what you get is in between, and bunch of that. So, this goes as a waste with the leaf, we call it the crown and the tip. The crown and tip is thrown away. So, my research group, we mash it, we make a pulp out of it, put it into a solid state reactor, run it just to see if we are able to extract the carotenoid, whatever is present in it. Now, carotenoid is a natural pigment that will give you the natural orange color, right? Now, other classifications of bioreactors are immobilized cell bioreactors and the fluidized bed bioreactors. We did discuss about the fluidized bed bioreactors a little bit. Immobilized cells bioreactors are where reutilizations of cells is required. So, your cells are immobilized into a different chamber, or you can say a flow cells or a flow cell, and then you have your substrate being pumped into that flow cell, and as it moves through the chamber, your cells are interacting with the substrate, your product is formed, it is pumped back, and the substrate, whatever conversion has taken place, is pumped back. So, it reduces the contact time, it increases the volumetric productivity. The fluidized bed reactors, you have your heat and mass transfer is efficient in this case because everything is fluidized. So, the transfer of temperature, the transfer of or the mass movement would be appropriate. Effective mixing between liquid, solids, and gaseous phases, and it has a very low shear rate, suitable for plant cells and mammalian cells. So, depending upon the type of host, so let us say if you are going to use mammalian cells as a host to grow something, you would like to go for a fluidized bed bioreactor. Okay. Now comes the main aspect of your bioreactor design and analysis. So, what are couple of parameters that we have to discuss here? Sorry. So, for the bioreactor design and analysis, till now we have talked about batch reactor, fed batch reactor, continuous stirred tank reactor, fluidized bed reactor, immobilized bed reactor. There is another type of bioreactor. Have you heard of this? Plug flow reactors. You know what a plug flow reactor is? Those who know it, please help me out. Okay. So, in the chamber, do you have the cells immobilized? Or what do you have in the chamber? Because that's a plug flow reactor, right? So, you have in my understanding, it is you're passing the substrate as a plug. So, it is moving from one side to the other side. It will get converted, or the substrate will get consumed and will go away, right? The product will be formed and that can come out. So, this particular chamber in a plug flow reactor, does that have your immobilized cells? Yes or no? In this particular design that I have made, yes. So, in a plug flow reactor, that is one way of passing it. It's a really nice design, but to be honest, I don't remember what exactly we use it for. I forgot. Dr. Bala?
A type of condensed reactor without any axial mixing.
Yes, there is no axial mixing. Axial mixing. So, in the axis there is no mixing. It's moving from one direction to the other direction, right? Okay. So, that's one other. So, plug flow reactors. That's another type of reactor which is there. Fluidized bed we have done. Immobilized cells bioreactors we have done. Okay. So, bioreactor design analysis. So, what size of reactor, type of reactor or method of operations are best for a given product formation? That's the very first question that you are going to ask yourself when you are trying to choose the type of bioreactor for your own process. Now, it depends. It very much depends for your own project, sorry, not project, for your own case studies, depending upon the type of product that I have asked you to make, you have to choose the right bioreactor. But that choice will depend on the host that you have chosen, correct? It will also depend on whether your product which is made is extracellular or intracellular. What else would be there? The host, the product, you need aeration or you don't need aeration. Do you need mixing, continuous mixing? What else would be there? Somebody give the answer. Environmental conditions. Okay, process parameters. What else would be there? Will the amount of product that you want to develop be a condition? That should be the primary condition, the productivity, right? Yes. If you're having difficulty, go back to this particular slide. Ease of operation, less downtime, product variability should be less. Productivity or yield is also a very important parameter in choosing the right reactor for your process. Maintenance is another thing. The skill requirements, I'm not going, don't consider that for your own case studies. That is not, that won't be necessary. Control systems, yes, would be very, very important. The type of feedstock that you are going to use will also define. Now, how do you define the performance? At the very beginning of our lecture, we talked about from the monetary perspective and the time perspective, right? If you have to say if a particular process is efficient enough, I will say how quickly I was able to get my product, or how much product I was able to get to make the money out of it. When you are looking at it from the reactor perspective, you will be looking at the reaction time. So, how much time it takes for the conversion of the substrate into the product. So, it is a measure of processing rate in a batch reactor, and so whenever we are working with a batch reactor, the performance measurement is based on the reaction time. When we are working with, let's say, a flow reactor, like a CSTR, continuous stirred tank reactor, we are looking at space time and space velocity. So, space time and space velocity. So, space time is the time required to process one reactor volume of feed, and the space velocity is the number of reactor volume treated in unit time to define the performance of a flow reactor. So, both space time and space velocity are used to define the performance of a flow reactor. Reaction time is only used to define the performance of a batch reactor. Before we go into the next set of slides, which will be later, I just wanted to go back on a few slides and have a bit of a discussion with respect to your case studies. That was the intent. Now, when we are looking at your case studies, so case study 1 was monolin. Please correct me if the spelling is correct or not. And the case study 2 was alpha amylase. For monolin and alpha amylase, for monolin it's a plant protein. It's a protein as a product. Now, what will impact the quality of the protein or the product in your fermentation system? Your pH will impact it, the temperature will impact it. So, the process parameters, right? So, you have to be very careful in terms of what reactor you will choose depending upon what will happen to my product at the very end. So, you want where you have maintained the temperature, okay. pH will change based on if the product is going to add to the medium. If my product is acidic or basic, that is going to change the medium pH. The protein, if that is extracellular, is it going to change the pH of the medium? Guess, yes or no? So, when you add protein to a media, will the pH of the media change, or any, let's say if you add protein in the water, does the water pH changes? You have your protein shakes, right, if you go to the gym. Does the pH of your protein shakes change when you mix water in the powder protein? So, let us say it does not. Let us say it does not. I am not agreeing with this or disagreeing with it. Just for the sake of this conversation, I am saying let us say it does not change the pH of your medium. The only parameter that remains would be temperature, right. If the temperature is also optimum, your interest is that you get maximum amount of protein generated, right. Then you can decide what type of bioreactor it would be. Be careful in that. In case study 2, alpha amylase, that is an enzyme, again a protein. Whether it is produced extracellularly or intracellularly, depending on the host that you choose, depending on the process that you choose, will define the type of reactor you are going to use. So you have to be very, very careful in choosing the right reactor design for these type of processes. The second part of our presentation is pretty long, the fed batch and the batch reactor, and this part was not too long. So, I would like to end the session here for this one, and we will meet again at 2 to start the second session. उनको बोलिए डरने के लिए मेरे पास बहुत मटेरियल है। हाँ Good evening. I hope you had a nice lunch. Okay, there are a couple of things that before we start the presentation, or that before we start the discussion on the design of the batch and the fed batch reactor, I have to clarify a mistake that I made in the previous session when we were talking about the plug flow reactor. I stated that the cells are immobilized and we are moving the substrate. That's wrong. We are actually sending the cell and the substrate as a plug. So, we send like specific concentration of the cell and the substrate through the reactor. So, it gets completely consumed, and by the time it comes out, it is like you have the whole cell mass, and then again you send the second plug, the third plug. It's a continuous plugging of that. So, it becomes like a continuous stir tank reactor where you have the continuous growth in it, right. Okay, so, now that I have fixed that mistake, I can start. So, design of the batch and the fed batch bioreactors. So, we were discussing about the design analysis. So, the idea is whenever you are trying to develop a bioreactor, or whenever you are trying to develop a particular process which is going to use a bioreactor, there are a couple of questions that you have to answer. So, one of the question would be the size of the reactor, or the type of the reactor, and the method of operation that you are going to choose, and which of those reactors and the method are better for your product formation. So, keeping in mind with respect to our case studies 1 and 2, you are actually going to go through this exercise yourself, but to give you a bit more information of how we actually design these reactors, how do we understand that this reactor is better for our product or that reactor is better for our product, we will get into the details of it. Now, we have also discussed about the performance measures. So, when we are looking at the reaction time, that is what is used for the batch reactors. If it is the space time, the space velocity, we are looking at the continuous flow reactor. So, that would be your continuous stirred tank reactors. So, batch reactor design, there has to be why we are doing it, right. So, when we are starting, the starting question for a batch reactor design would be like for a given reaction kinetics and the desired substrate conversion, what should be the volume of the reactor to meet the production target. Now, for a batch reactor, now if I have to give you the most layman answer, let us say if you have to cook rice for yourself, how many glasses of rice will you put in the pressure cooker? And if you let us say you have a party of 4 or 5 coming to your house, how many glasses of rice will you put in the pressure cooker? Will a 2-liter pressure cooker be good enough for you? Whether a 5-liter pressure cooker would be good enough for you? That sort of question is what we are trying to ask. Okay? So, what would be the size of the reactor? If we want a specific desired level of product formation or the cell mass concentration, what should be the size or the volume of the reactor that we should be using as part of the batch reactor? The other part would be to calculate the productivity of the given reactor volume and the process kinetics and the conversions. So, these are the 2 questions that we would like to answer as part of our batch reactor design. The other, how will we do that? So, the way we will go forward with that would be that we have to develop an equation between the cell mass generation, which is going to be denoted as X, the product that we are generating would be P, and the substrate that we are consuming as an S. So, we have to find the relationship between the cell mass, the product and the substrate. So, there is a relationship between the cell mass and the substrate. There has to be a relationship between the cell mass and the product generation. Sometimes, if you have too much of substrate, that is not good for your cell mass. Too less of substrate is also not good for your cell mass. If there is a substrate inhibition, what would be the situation? If there is a product inhibition, what is the situation? So, we will be looking at all those type of equations or relationships in terms of the batch reactor design. Now, these equations are used to predict the batch time for a desired conversion. So, once we have understood the relationship between the parameters, the cell, the product and the substrate, we can use that to understand how much time will it take to get the specific product or the specific cell concentration using the specific amount of substrate or given volume of the reactor. And we will also be able to estimate the volumetric productivity of the system through our batch reactor design. Now, the growth kinetics. The growth kinetics, or when we are talking about the cell growth, if you remember in the very first conversation that we had on the cellular agriculture, we did talk about the sigmoid curve, right. The cell has a growth phase, lag phase, the log phase and a stationary phase, and then you also have a death phase eventually, right. So, that is basically your cell concentration x with respect to the time, you are looking at how the cell mass is growing. Now, how do you actually quantify this cell concentration? So, there are 2 methods. You have the direct methods in which you are looking at no suspended solids or interference compounds, and then there is a preferred method which is actually your dry weight value. So, if you remember when you are doing experimentations as part of your microbiology course, if you are growing a cell in a media in a shaker flask, you will be continuously taking out at least an ml of the media and checking its turbidity, and that is what you are going to plot to check your growth curve, right. So, that is what we do, the optical density. So, if you can take the dry weight and optical density, which is estimated between 600 nanometers to 700 nanometers, or you can use the cell number density, or you can estimate the cell number density using hemocytometer or a plate count or a Coulter counter. So, using these techniques, you can actually estimate how much of the cell has grown in your medium. Now, the indirect methods are preferred when the direct methods are inapplicable. That would be specifically for the solid state mold fermentation. So, solid state fermentation technique is widely used when you are trying to grow mycelium or fungus for your own product as a product. So, solid state fermentation system is used. So, we basically use the indirect methods for that. So, the cell mass can be determined indirectly by measurement of the protein or the DNA or the ATP or the NADH. Now, if you are looking at the ATP and NADH, these are products of the metabolic pathways, right, either glycolysis cycle or the TCA cycle, you do get these produced. So, if you are able to, if you are aware of how much of ATP would be generated by the biomass or by a given biomass, you can estimate by estimating the total ATP amount. So, for example, the example that is given to you is like, assume that 1 milligram of ATP per gram dry weight of bacterial cell is produced. So, if you have 100 milligrams of ATP per liter, then you can estimate your cell mass concentration to be 100 grams of dry weight cells per liter. So, you are estimating it indirectly how much of the cell mass is there by estimating a product that has been generated out of the cell or within the cell as part of its metabolic pathway. Or even if the protein is being produced, how much protein is being produced per cell, if you know that, then you can go back and estimate how much your cell concentration would be. So, you have the direct methods. So, direct method is the turbidity method, that is the preferred method. The indirect method would be when we are looking at the DNA, the ATP, NL and the NADH. Now, with respect to the design and operation of the fermentation system, the cell growth kinetics plays a very important role, and understanding the cell growth kinetic is also very, very important. So, cell growth kinetics is the rate of cell growth and how it is affected by the various chemical and the physical conditions. Now, when we are talking about the chemical and the physical conditions would be, if you can give me an example of physical condition. Use the mic, if you can use the mic, that would be really great.
Like this pH and these environmental conditions.
The chemical would be that.
Okay, okay, physical.
What would be the physical? See, the change in temperature, change in pH will affect your growth kinetics for sure, right. So, that is where we are looking at most of the time. So, when we are looking at the cell concentration with the change in the cell concentration with respect to the temperature and the pH. My question to you was like, if when we are talking about various chemicals and the physical conditions, what would be one of the physical conditions? That would be the temperature. Chemical conditions, pH. We can take that way, right. Now, when we are having the growth kinetics, as we said, like the growth curve looks more or less like this. You have your death phase at the very end, you have your lag phase, this is your log or the exponential phase, and this is your stationary phase. There has to be a mathematical equation that can actually define this relationship, right, with respect to time, the change in concentration of the cells or the x with respect to time. There has to be something. Now, how do you accurately model this, mathematically model the cell growth? It is actually very, very difficult to do that, because the growth is dependent on various biochemical reactions and transport phenomena. So, let us say if your mixing is not proper and it is not able to get enough oxygen or the nutrients, the cell growth will vary at different parts of the reactor. If it is a batch reactor, it is changing at very different parts of the reactor. So, mathematically you have to assume that there is a homogeneity, the completely homogeneous environment is maintained, there is no difference in the nutrient content on the top and the bottom. So, you have to assume a lot of it to be able to mathematically design an equation that can define your growth kinetics. A heterogeneous mixture of young and old cells are present at all times. If you have this type of scenario, your growth kinetics cannot be modeled mathematically, because younger ones will have a different model, the older ones will are already at the stationary or the death phase. It changes because you have a mix of it. So, the mathematical models that are there which can actually explain cell growth fall under the unstructured and distributed empirical models. Now, there are certain assumptions that have been made for these empirical models, and we did discuss about these assumptions in the previous slide. So, assumptions concerning cell components and the population, that as the cell is increasing, the cell components are also increasing at the same time. The cell kinetic equations derived from these models are used for the analysis and design of ideal fermenters, and that because whenever we work on the design of bioreactors or design of fermenter systems, whether we are going to do the batch or the fed batch, we are always going to design for an ideal batch reactor or an ideal fed batch reactor, or even an ideal CSTR if you go up to there. So, the assumptions are the cells represented by a single component such as the cell weight or the cell number, assuming balanced growth. That means, the cell weight and the cell number is increasing at the same rate as its components. The second assumption is the cell suspension is homogeneous, uniformly distributed. Okay, the nutrients have been uniformly distributed. If the oxygen is being added to the bioreactor, it is uniformly distributed. So, all the parameters that are required for the cell to grow are uniformly distributed, but when we are only considering the cell. So, the cell are homogeneously distributed. Only one medium component is limiting the reaction rate, other components are in excess. Now, when we talk about the limiting component for a cell growth, what would they be? What does a cell require to grow? Just give me two. In the mic, that would be great.
Sir, carbon and nitrogen source.
Those are your nutrients, right?
Nutrient.
So, nutrient or the substrate that is coming in is one.
Yes, sir.
What is the next one? What can inhibit a cell growth? What can change the cell growth?
Oxygen.
Oxygen, right? So, you have your nutrient concentration and the oxygen concentration. So, if you change either of it, you will be varying the way the cell is going to behave, or the cell growth is going to behave in the fermentation system. In this case, we are assuming all of the other things are constant, only one is changing. Either we are changing the substrate or we are changing the oxygen. Majority of times, we are changing the substrate. The substrate is your limiting factor if it has to be. The concentration of the substrate that is going to be added to the reactor would be your limiting factor. Other than that, nothing else changes, because we have defined, because when I say that homogeneous nutrient, the homogeneous distribution of the cell, homogeneous distribution of the nutrient, homogeneous distribution of the oxygen for the cell growth, I am only changing the nutrients, the rest all remains the same. So, monod model is one of those empirical models which is commonly used to define your cell growth. Now, in this particular case, you have mu, which is your specific growth rate, So, your mu is equal to mu max S over S plus K S. Now, this is your Monod's saturation constant. This is your limiting factor, because we have to make that assumption of the limiting factor, right. So, this is your limiting factor in this case. So, Monod's model, the S over K S plus S is your limiting factor. Now, how do you define your mu, your specific growth rate? Dx by dt would be the change in the cell concentration with respect to time, right. If you do normalize it with respect to the cell concentration x, that is what is going to give you your mu, the specific. So, mass of cell produced divided by the original mass of cell into time. So, if you rewrite this equation, that is what has been done in the last line here. So, dx by dt becomes mu x or mu x naught is your initial cell concentration, and we know that mu is mu max S over K S plus S, you just substitute that. So, this is the equation that you will get. So, further increase in the nutrient concentration, mu after it reaches max does not affect the mu. So, what it means is, as you increase the S, or S tends to go to infinity, just for now I am just keep on increasing the S, the cell concentration will increase and it will become flattened out. This is where your mu max is. And if you remember your Michaelis-Menten constant from your enzyme kinetics, when we are doing that, so the half of your enzymes, the same thing is used here. So, the half of mu max by 2 is your K S, the constant, the Monod saturation constant. Okay. Clear till now? Any questions? These are the basics. If you have already done that, I am sorry, I am going to repeat it anyway. Now, this particular graph here is representing your growth kinetics. So, you have your lag phase, you have your acceleration phase, the growth phase, and the decline that goes into your stationary, and then you have your death phase. Just a question for you, when the cell is in its lag phase, what does that mean? Use the mic, please. If you have the mic, please answer in that.
Let's try to.
It's on. It's on.
Let's try to adjust to that environment.
It is trying to adjust to the environment.
Adjust to
right?
the environment.
Why is it trying to adjust to the environment?
It has to utilize the carbon source. It will take some time.
But if I remember correctly, when we were discussing about the fermentation system, so we have a seed fermenter where we have grown the biomass. I have acclimatized it to the nutrients that are going to be used in the larger biomass. Will it still require that lag phase?
We can reduce the lag phase timing. So,
if you are using a seed culture, you will
we can reduce the lag phase time.
phase. So, if you use a seed culture, many a times this particular lag phase will not even exist. So, when you do not use it, it is obvious because the cell is growing, cell is still growing, it is not doubling, it is just growing in volume. Okay? The cell is still growing, it is not doubling, it is not multiplying. What it is doing is that it is changing the internal machinery to get used to the nutrients in the environment that you have put it into. Once it get used to that, it knows now I am ready, let us double. That is where your acceleration phase starts, and then gets into your exponential phase of cell growth. Once it is in the exponential phase of cell growth, it will remain in the exponential phase of cell growth if you continuously provide it with substrate. Am I right with this statement? If I continue to provide substrate, will the cell continue to be in its exponential growth phase? I am not saying a cell, I am talking about the whole reactor. If I continuously keep on adding the substrate, can I maintain the exponential phase for as long as I want? No? So, you won't be able to maintain the growth phase at all, or the exponential phase, the log phase? Okay, when will the cell get into the stationary phase? Use the mic, please. When will the cell get into the stationary phase?
Substrate limitation, or the cell will produce toxic components.
Okay, let's assume the cell is not producing any toxic components.
Okay.
And there is no substrate limitation.
Okay.
Will the cell reach its stationary phase?
Sure.
Like, do cells have a biological clock like humans?
Maybe.
They have a life cycle, like, okay, I am only going to be alive for 1 hour or 2 hours, or they will continue to keep on growing and multiply. You are talking about bacteria. Will the cell biomass keep on increasing? That is what my question is. I am not asking you anything else. Will my cell concentration be in an exponential phase or continue to grow if I continue to provide it with substrate? In a batch reactor, that is not the situation, right? In a batch reactor, you have given it enough substrate, you have given the seed, the substrate, close, done. You let it grow. Once the substrate is completely consumed, it has been in the stationary phase. Like it has reached the max that it can be in that mu max stage, and once it has completely consumed your substrate, what will happen? Then your decline, your death phase will start, but before the death phase, the cell will still catabolize itself to grow. It will kill other cells to get the component for it to grow. We will be learning more about that later. But I am just, remember this question that I asked you. If you continue to provide the substrate, will the cell continue to grow or not? Just remember this question, okay? You will be able to answer it later on. Now, let us get into the Monod's equation. So, there are different, so Monod equation is with respect to your cell growth, right, cell kinetics. There are different versions of Monod equation that have been exist. I think this is your Luong model. This is to represent the substrate inhibition. If the substrate is the one which is inhibiting the cell growth itself, how do you represent that? You will be representing it with this equation. It's a upgraded version of your Monod's model, and this is the only part which has been added, because what is your mu is equal to mu max S K S plus S, right? That is what your Monod's model is. If you add this 1 minus S minus S m to the power A, you are adding the substrate inhibition aspect. So, this is your inhibition from the substrate side. Now, when the substrate inhibition takes place, so let us say if I have an equation with mu and substrate. So, there is, let's make a graph. So, when we are talking about inhibition, there are two ways the mu will behave with respect to the substrate concentration. Either it will follow an asymptotic curve, or it will follow a straight line, a declining straight line, or with a negative slope. So, when you are looking at this declining straight line with a negative slope, the location where it has intersected the substrate line, the x axis, that is your SM. So, what it means is the Luong model is valid if you are provided with the concentration of the substrate that is actually going to inhibit your growth, your mu. So, if you are aware of the concentration of the substrate at which my growth will get inhibited, you follow the Luong's model. If you are not aware of what my substrate concentration is going to be at which the inhibition will take place, that means your mu will become 0. You do not use the Luong's model, you use the second one. The relationship, the asymptotic relationship between mu and S is represented by the equation 2. If this is your equation 1, that one is only valid if you are aware or if you are given with the value of the substrate concentration that will inhibit your growth, or your mu will become 0. Now, if multiple substrates are present, an upgraded version of the same model would be used. Seba? Now, if you are looking at product inhibition, so I have substrate inhibition. Sometimes if you have growth of product, product might also inhibit the cell growth, right? Can you give me an example to that?
Ethanol, sir.
If you have more ethanol generated, it will inhibit the cell growth eventually, right? So, if the product is the one which is going to inhibit, I am going to ask you a simple question: which of these, if I am looking at mu and the p, which of these will follow the negative slope or the asymptotic behavior? 1 or 2? So, which one will follow the negative slope? So, you know the concentration of your product at which your inhibition will take place. So, your number 2 is a straight line and your number 1 is going to give you the asymptotic behavior. Same equations, similar equations for the substrate and the product. These are the upgraded versions of Monod kinetics or Monod's model. So, Monod's model is used to define your growth kinetics. If you have inhibitions associated with it, you have to use the Monod's model, but a different upgraded version of it, and that is what they did. Any questions? Now, let us come back to our discussion on the different phases of the growth of in the batch culture. So, you had your lag phase. So, we did discuss that the cell is going through the machinery changes, right, but it will increase in the volume, but it is not going to increase in number. In the acceleration phase and the growth phase, it is going to increase in the number, it is going to multiply. Once it has reached the decline or the stationary phase, that is the time when the relationship has become that it is about to finish off all the substrate that is available, right. So, the balance has been reached, and once all the substrates are gone, what happens? Death. And that is where I did state that it might start eating itself, right, not cannibalism, catabolism. Cell catabolism starts to take place, or endogenous I think I forgot it. I think I have it that in the slides, so we'll go there. So this is your cell growth kinetics. We did discuss a bit about your lags, the growth, and the stationary phase. Now, do cells produce products as they are growing? So, in their growth phase, what sort of product will a cell produce? Primary metabolites. Secondary metabolites comes in the stationary phase. Your antibiotics, vitamins, minerals, they come out in stationary phase. In the growth phase, it's mostly the proteins and anything that is required for its own growth, right? So, anything which is within the cellular structure of the biomass, like the cell. So, lag phase is a period of adaptation. We are just going to repeat a little bit for the cells to their new environment. Thank you for answering that question at that time. So, new enzymes are synthesized, a slight increase in cell mass and volume, but no increase in the cell number. There is a prolonged, it can be prolonged, you can prolong your lag phase if you have put less inoculum. You can do that. You can also prolong your lag phase if you have a poor inoculum. That means either you have like a mix of dead cells with your viable cells. It's like it's an old culture if you take that. The age of inoculum is older, then you are going to prolong your lag phase, or your medium in which you have put your seed culture is not nutrient enough. So, it is going to remain in the lag phase for a longer period of time. So, if the medium contains more than one or more carbon sources, you will be observing multiple lag phases, and that is called as dioxic growth. Now, what happens in that? We did discuss about it. So, let's say if you're in the recombinant DNA technology, when we are talking about the, if you remember, for one of the examples, there were two plasmids that were added into the cell, into the bacterial cell. I don't remember exactly which example was that. If I think it was PET. So, when it was being grown in using the cell, there were two different plasmids. Let's say there were two different plasmids. Now, one plasmid happily works or will make the modifications to the cell in such a way that it can use one carbon source. The second one requires a different carbon source, or there are two different carbon sources that are present in the cell. Forget about the plasmid for a moment. So, if there are two different carbon sources, which one will the cell choose first? I asked that question, right, C1 and C2. It will choose the one that it likes the most, because everybody wants to be in their own comfort zone. Cell also wants to be in its own comfort zone. So, it is going to choose the one which it likes the most. So, you will have a growth with a small lag, you will have the growth. Once it had completely consumed that particular carbon source, now it has to rearrange its metabolic pathways to start consuming the second carbon source, which it might not like, but it has to eat. It's like when you go to the hostel, you might not like the food, but you have to eat it, right, for survival. So, that's exactly what happens. So, you have, that is called your dioxic growth. So, when you are using multiple carbon sources, it will continue to use the carbon source that it likes the most, then the second one, then the third one. If you keep on adding, you can keep on having that growth, multiple lag phases. The exponential growth in this particular phase, the cell has adjusted to their new environment, and it will start multiplying rapidly, exponentially. Okay. So, it does a balanced growth. All components of the cell grow at the same time, and the growth rate is independent of nutrient concentration as the nutrients are in excess. The growth rate is independent of the nutrient concentration for the exponential phase, because the nutrient present is in excess. So, I will ask my question again, what if I maintain my nutrient concentration in excess? Will my cell remain in the exponential growth? I am not asking you to answer right now. It might be a trick question. So, think wisely whenever you get the idea to answer this, think wisely. If I maintain that excessive nutrient concentration, will my cell remain in the exponential growth phase? Now, before we get into the design equations for the batch reactor and the fed batch reactor, we have to go through certain terminologies, or we have to understand certain terminologies which will be used more or less every time we try to solve. So, in our conversation on the cell biomass and when we are looking at the microbial growth, we will be using a terminology called as Cx, which is your cell concentration, is your dry cell weight per unit volume. Your dCx by dt is your change in your cell concentration with respect to time. So, when we try to do, if you remember mass balance, right, in minus out plus generation minus consumption is equal to accumulation. Your dCx by dt is your accumulation term. In a batch reactor, do you have an in? Everything is already inside, right? There is no inlet, there is no outlet. Is there any consumption? Consumption of what? But am I balancing substrate or am I balancing the cell? Am I consuming the cell? So, the only thing that you will have is generation, right. So, let us say that I will give as R x, but it is a volumetric generation, volumetric accumulation, because the V remains constant. Got the idea? This is actually your design equation for your batch reactor. I have given you way in advance, but I am just trying to explain how we are going to use these terminologies later on. Okay. So, your R x is your growth rate of the cell biomass. So, what is the difference between dCx by dt and R x? dCx by dt is a change in the cell concentration, R x is your growth rate. The difference between these two is R x is 1 by x dCx by dt. You have to have that term of x, because the rate of growth. So, it will appear that both the terms 1 like the 2 and the 3 are the same, but the 2 may include the effect of input and output flow rates and the cell cycle, because you might do a cell cycle, it might keep on increasing it. Now, these terminologies can be used for batch reactor, it can be used for fed batch reactor, it can be used for CSTR. These are not specific to batch only, but these are the terminologies that are basically used when we are going to go forward in our conversation here. 3 is your actual growth rate of the cell. 3, which is your R x, is your actual growth rate of the cell. 2 is only equal to 3 for the batch operations. 2 is only equal to 3 for your batch operations. Now, the other terminology would be that growth rates based on cell number and cell weight can be assumed to be equal in exponential growth phases, with a balanced growth assumption that all the components are also growing at the same time. And the microbial growth, the product formation and the substrate utilization rates are usually expressed in the form of specific rates. So, the growth would be represented as mu, which is specific growth rate. The substrate will also be represented similarly in the specific. We will have the product, you will see QP for the product or the specific growth of the product. We are always going to represent it in terms of the specific rate. Now, specific rate are always normalized with respect to the initial concentration. So, that x has to always be there for specific growth. So, what was the equation for mu? That is your specific growth, the change in concentration with respect to the initial concentration multiplied by time. There is another terminology which is your division rate. Now, we all understand. So, if let us say a particular cell is going to divide it into 2 at a time, right. So, if let us say your cell, if C naught is your cells dividing n times in time t, the total number of cells C n after time t would be so your average division rate delta is your n over t. Now, this is the equation that comes with from the average division rate. Now, if you keep on rearranging the previous equations that we have been looking at, what you will end up with is this. This is the most important equation in this whole list of equations here. So, your R n or your R x was dx by dt, right. Or your dCn in this case. So, dCn by dt, and they have done a ln of it and ln of the n values, and they have just rearranged it and put it back into the equation for mu. So, the division rate is constant in exponential phase, while growth rate is not. What it means is, as you are in your, how do I go back? So, in this growth phase, the division rate is constant. Okay? Growth rate is not constant. The slope of this is your constant n. The other terminology that we are going to use would be your doubling time. So, your dx by dt is given as mu x, or mu is 1 by x dx by dt, right. So, this is what the equation is, where your mu is your specific growth rate, x is your concentration in grams per liter, time is the time in hours, as you are rearranging it. So, if you make it dx by x mu dt, and if I integrate it, I will be left with mu t, like 0 to t, this would be x naught to x, this will become ln x by x naught is equal to mu t, right. That is what they are doing. So, this equation, you have your doubling time, right. So, when your x becomes 2 times of x naught, that is where your t becomes T d. So, the equation for the T d is ln 2 by mu. So, your T d, your doubling time, is basically a reciprocal of division rate. This is a reciprocal of your division rate. So, growth rate and the division rate defined by the cell numbers are different entities. The division rate is constant during the exponential period, while growth is not. This is the most important concept that you have to take into account. Division rate is constant during the exponential period, while the growth is not. Now, exponential growth phase, the balanced cell of the cell mass in the culture is given as excuse me. So, the balance of the cell mass in the batch culture gives is the dx by dt is mu x. X is x naught at time t is equal to 0 or when it is starting, and the integration of this is given as your dx by x mu t, right, sorry, mu dt, sorry, my mistake, mu dt 0 to t x naught to x. So, ln x by x naught is become mu t. Now, if you put the equation of x naught is e to the power mu t. If I give you this equation, let us say if I give you the initial concentration that came into the batch reactor and I give you the time of the total batch or the batch time, you would be able to use this equation given that I have provided you a specific growth rate. If I provide you all of those parameters, you would be able to tell me what the cell mass concentration would be at the end of the time t with the given specific growth rate. You will see that we will be using this equation to solve a few questions later on. Okay? So, this particular equation is an important equation in terms of your batch growth kinetics and batch operations. This is what we are looking at mostly in the exponential phase, right. The slope of this would be constant. This is a constant slope. So, the mu remains constant in this phase, in the exponential phase, it remains constant. Now, stationary and the death phase, exhaustion of the nutrients, that means your S has been consumed completely and the buildup of the waste and the secondary metabolic products inhibits the growth. The cells may have active metabolism to produce secondary metabolites, that is where in the stationary phase it happens. Endogenous metabolism occurs by catabolizing cellular reserves for new building blocks and energy producing monomers for the maintenance energy. So, that means the old cells or the dying cells will be providing the materials for maintenance of whatever cells are remaining or that are still there. Okay. The dying cells or the dead cells or the ended cells are the ones which are providing the maintenance materials. The cell lysis may occur and the viable cell mass will drop in the stationary and the death phase. So, the growth kinetics with respect to your stationary phase is given with, instead of mu, we have this minus k d. This minus k d is the rate constant for endogenous metabolism, because now the growth is endogenous in nature to catabolism. We are looking at k d rather than mu, which is your specific growth rate. It is related to specific growth rate as mu net, because you have your mu and then you have your k d. So, total mu net would be mu minus k d, or sorry, mu into minus k d, where your mu is your gross specific growth rate. So, endogenous metabolism is the cell catabolizes cellular reserves for new building blocks and for energy producing monomers, which are used for maintenance, okay, which is used for maintenance. Now, let us get into other some of the terminologies, which are your yield coefficients. So, yield coefficients are always represented as with y. So, it can be y x by s, this will represent the yield of x with respect to S. So, that means the cell with respect to substrate. You will have y p by S production of product with respect to substrate, and y x over O2 with respect to how much O2 is consumed. Now, y x by S simple equation would be x minus x naught divided by S naught minus S. S naught is your initial substrate concentration and S is your final substrate concentration. X naught is your initial cell concentration, X is your final cell concentration. So, the ratio of those two will provide you your yield. Now, majority of the slides that I am using for your batch analysis, these were taken from an NPTEL course. I remember Dr. Smita Srivastava from IIT Madras, because when I saw that same presentation, I teach the same course back in Canada. It was pretty much the same thing that I wanted to teach. So, I used more or less her slides or her concepts, but I have added my own flavors to it, the way I explained. That would be it. It reduces the workload, but I would like to acknowledge her and thank you for providing these slides. Okay, so yield is clear, how we write it, the notations of it. We will get into how the equation works out. You will be seeing how we will be rearranging the equations. So, if you have to have x, if you know the yield, let us say, if you are provided with the yield, you have your y x by S, right, multiplied by S naught by S plus x naught. So, let us say if I give you the initial cell concentration, I give you what the yield of the cell biomass is and what was the substrate concentration before and what remained after. So, let us say I am asking you what would be the cell concentration after 70% of your substrate has been used, if the substrate that was being put was 10 milligrams or 10 grams per liter, or this much of the substrate was added. You would be able to use the same rearranged equation to estimate the amount of x. Okay. Now, these are for your batch process, only for batch, not fed batch, batch. Batch growth kinetics for most bacteria and the yeast, the yield y x by S for glucose, it ranges between 0.4 and 0.6. This is in your Schuller and Cargi textbook, okay, which is like the Bible for bioprocessing. Similarly, y x by O2 would be 0.9 to 1.4 grams per gram O2. So, how much cell is produced by how much oxygen is being consumed. So, during the batch growth, the measured yields are apparent due to endogenous metabolism occurring, that is the Kd is greater than 0. So, let us say what it means is your cell grew, reached the stationary phase, right. In this stationary phase, we had this conversation that the cell will maintain by killing other cells, or other cells will lyse and whatever content of that cell came out can be used by the other cells to keep themselves alive. You are getting my point, right? That, if that is higher, that means your Kd, instead of that mu, if the Kd term is higher or greater than 0, the yield that you are getting at the very end of the batch reactor, that means I have done everything. So, let us say if this is my reactor, I put up everything, I was supposed to open it after 1 hour. I got delayed by 1 hour, let us say by 5 minutes, and I opened it and I took out everything. My yield will not be the same. You getting my point? My apparent, the actual yield that I will get is different than my theoretical yield that I would have calculated. Okay? So, my yield varies depending upon if my endogenous metabolism took place, because some of my cells are gone for the maintenance of whatever remained, because I was delayed by, let us say, 5 minutes. That is the best layman way of understanding it, because it has finished all the substrate. Now, the cells are like, okay, we are done, take us out. I didn't. They kept on knocking, I didn't. After 3rd minute, they are like, dude, we have to survive, let us kill people. Endogenous metabolism takes place, my cell concentration is going to go down a little bit, right? My yield will reduce little bit, very little, but not significantly, very little. If my KD is way too high, it will be a significant decrease. You got the idea how it looks like. So, at the end of the batch fermentation, we have apparent growth yield, which can change with culture conditions. So, it is not truly constant. Now, comes the design equation. So, you remember when we were doing the accumulation, input, output, generation, consumption. So, no consumption, because we are balancing the cells. There is a difference between heat energy balance and material balance. In material balance, you always balance each and every component of a mixture. Your cell in a suspension is one of the component in a mixture. So, you are going to only either balance your cell concentration or your substrate concentration or your product concentration. You cannot balance all of them at the same time. You can only balance one component at a time. So, one mass balance equation for one component, clear? That is the reason why we are only using the cell concentration here, because that is what our interest is. We have already done this. So, dx by dt will end up to being so v dx by dt becomes v rx. Rx is mu x. So, Rx is your mu x. So, now we know dx by dt is mu x. Now, mu using the Monod's model is mu max s plus ks into x, right. So, this is the equation, that is how it came. So, your Monod's model came up here for the value of mu. In the batch, the nutrients are added at the beginning and the product is withdrawn at the very end. The cell balance equation in a batch reactor is, oh, we have already, oh sorry, there is two of the same thing. My mistake. So, your dx by dt is your mu x, okay. We have already estimated that. We substituted mu to your mu x s divided by k plus s plus ks. You remember this equation? Yx by s is x minus x naught divided by s naught minus s. So, x can be written as y x by s, s naught minus s plus x naught. So, that is your equation. Clear? You know your mu. So, we are putting both of these equations down there and then we are integrating it. So, when you integrate it, the equation that you will get is this. This is called your design equation. So, it is T mu max A ln of x over x naught plus B ln of s over s naught. This is your design equation, where your A is Ks Yx by S divided by x naught plus S naught Yx by S plus 1. Similarly, your B is given. So, these are the two equations that are used, but this is what is called your design equation for the batch reactor problem. Now, we have discussed about the growth kinetics. Now, let us discuss about the product kinetics. Now, let us say, sorry. So, your growth associated products are the products produced simultaneously with growth. Example are constitutive enzymes. Now, what are these constitutive enzymes? Are the enzymes that are produced by the cell for their own growth. Okay. These are the ones which are produced during your exponential growth phase. In the non-growth associated product formation, that takes place in the stationary phase, because there is no growth taking place, it is stationary. That time your QP, now what is your QP? Specific product formation rate, right? Specific growth rate was mu, QP is a specific product formation rate. I did give you a little bit of a hint on that. So, let us say go into this. So, if your P is your product, so your dP by dt is a change in product concentration with respect to time. So, your QP would be 1 by x dP by dt. Now, what is your Yp by x? So, Yp by x is a change in the product concentration with respect to, yield of product with respect to the cell concentration. So, that means change in product or del P over del x. I can rewrite your Yp by x as dP by dt dx by dt. Now, let us say this is your equation 1. We know mu is 1 by x dP by dt. This is your equation number 2. Using both your equation 1 and 2, you will get QP, is mu y p by x, or 1 by x dP by dt. You do not have to write all of these things, it is just for you to understand how we are looking at. So, there is a specific product formation rate, there is a specific growth rate, mu, QP is your specific product formation rate. So, the equation that we have just developed is this for the growth. Similarly, for the non-growth associated, we are going to have the QP, which is equal to constant, which is beta here, and during the non-growth associated product, non-growth associated phase, non-growth associated product formation would be your secondary metabolites. The mixed growth product formation takes place during the growth and the stationary phase, and that would be your example of lactic acid fermentation and some of the secondary metabolites that are being produced commercially. If you are looking at from the commercial perspective, we try to be in the mixed growth phase if you are looking at both. Now, this is how your growth associated product formation, the mixed growth associated product formation and the non-growth associated product formation looks like. The primary metabolites are your growth related, example ethanol by Saccharomyces cerevisiae. The secondary metabolites are non-growth related like antibiotics and the pigments. So, if you are looking at the natural color being produced, you are looking at it in the non-growth associated phase. If you are looking at the antibiotic production, it is in the stationary phase again. And this is where your mixed growth, this is non-growth, and this is where your mixed growth phase takes place. performance equations for the batch fermenter. Again, we are just going to go through the equations very quickly. These are the equations that you are going to use when we are going to solve a few questions. So, your dCx by dt is your Rx or mu Cx, because Rx is mu x, right. We have already done this. Same thing, what we are doing is rearranging the thing. So, dCx by Rx, I will have mu dt. Now, if I sorry, no. I will just have the dt. Now, you integrate it, your T naught to T, x naught to x or Cx to Cx naught, you will end up with this equation here. Okay. Now, if you take this equation and let us say in this case, if I plot 1 by Rx over Cx. So, 1 by Rx over Cx. I am just looking at this particular equation here. So, if I take that and I am plotting this 1 by Rx over Cx, I will get this is where sorry, it comes here. So, this is where I have harvested my cell biomass. This is the concentration at which I have harvested my cell biomass. So, this is my concentration. So, this is my Cx and this is my Cx naught. The shaded region under this particular graph is your batch time. Or the batch growth time is the area under the curve. Now, T naught is your starting time. So, if you have had a lag phase, there would be a value for T naught. If you have not had any lag phase, if you had your seed culture and it started growing immediately, it will be 0. Okay. So, let us say you have your substrate which is being consumed by the x, you get your x, and that x is also giving you another x. So, x is basically being the catalyst here. So, the rate of reaction is slow at the start of the concentration of x. As the concentration of x increases, the rate of reaction increases. So, it increases as cell multiply and reaches a maximum rate. So, as the substrate deplete, the toxic products accumulate and it decreases to a low value. This is what we have already discussed. So, you have 1 by Rx Cx. This shaded area is basically your batch growth time, and it is useful for comparing the performance of various fermenter types. So, if you are looking at the batch reactor, the fed batch reactor, the CSTRs, the plug flow reactors, you can actually develop this equation for that. And when you compare how much would be the shaded areas for all of those fermenters, you would be able to say this one is better or that one is better. These are some final slides, and then we get into a couple of other parameters. You want to take a break? We'll continue for some time and then we'll take the break, and then we have to get into fed batch. Okay, so performance of the equations for the batch fermenter. So, Monod's parameter cannot be estimated by series of batch runs as done for Michaelis-Menten constant parameters. So, initial rate of reaction is always 0 for cell cultivation, unlike enzyme reaction. So, there is an extra Cx term because when you are looking at this, this is what they are talking about. So, when you are looking at the cell and when you are looking at the product, the difference, this is the Cx term which has to have, which is always there, because mu is 1 by x dx by dt, right. So, that is where it will come. So, that x goes back to the Cx is that 1 by x, 1 by Cx on this side. So, that term always exists, and because of that it is can never be 0. So, it is like you will always get, if I remember correctly, the other way around, wait. I am not going to draw the graph because I might make a mistake in there, but anyways. Okay, so, effect of temperature on the growth kinetics, we will just skip the slides pretty quickly. These are some things that we have already discussed. So, optimum medium pH, temperature, oxygen supply differs for various microorganisms. So, every microorganism will have different requirements for temperature, pH and oxygen demand. So, according to optimum temperature for the growth can be classified for bacteria. If you are looking at the psychrophiles, the optimum temperature is less than 20 degrees Celsius. For mesophiles, it is anywhere between 20 degrees Celsius and 50 degrees Celsius. For thermophiles, it has to be greater than 50 degrees Celsius. So, if you choose a thermophile for your product formation, you need to maintain a fermentation temperature of greater than 50 degrees Celsius. So, above the optimum temperature, the growth rate decreases and the thermal death starts to occur. So, just like that endogenous catabolism that we were talking about, we will have thermal death that will take place. Your mu net will change. So, the growth rate increases to a maximum with an increase in temperature when it is T optimum. So, what you will have is, if you remember in the morning I did draw this. So, at particular stage you have your T optimum temperature, your growth will continue to increase. Once you cross the T optimum in your temperature profile, your growth is gone, your cells will start to die. So, at a higher temperature, thermal death rate exceeds growth rate, which can cause net decrease in concentration of your viable cells. And the equations that can be used is, this is for your the thermal growth, AE, Arrhenius equations have to be used here. So, AE to the power e to the power a minus RT. So, when it is EA, it is thermal growth. If it is ED, it is death. That is a big difference between these two. So, temperature plays a very important role when you are looking at batch or any of the fermentation processes. Now, product formation and the yield coefficients are also affected by temperature. As the temperature increases beyond optimum value, the maintenance requirements, that means the coefficients of the culture increases. As a result, the yield will decrease. If you remember, we already had that Yx by S, the apparent yield and the actual theoretical yield. So, diffusional rate should be considered, which might become rate limiting step as the bioreaction rate increases with temperature. Now, temperature also changes something in the bioreactors. What will the temperature increase due to the, let us say, the viscosity of the medium? It will change the viscosity of the medium. If the viscosity of the medium changes, it is also going to change the mass transfer coefficients of how oxygen is going to diffuse, how the nutrients are going to diffuse. So, in most fermentation, pH can vary substantially depending on the nature of the nitrogen source, production or utilization of the organic acids, and the evolutions or the supply of CO2. By changing the CO2, the pH is also getting affected. And increase in pH, it will affect the activities of the enzymes, and therefore, it will affect the growth rate. Now, DO, which is your dissolved oxygen, is an important substrate in aerobic fermentation and may be limiting substrate as it is sparingly soluble in water. Do you remember, I'm not sure if you, in mass transfer, you would have studied something called as Henry's constant, right? That deals with dissolving gas into liquid at equilibrium, right? In a fermenter, in the fermentation system, can you change the pressure? Because changing the pressure will change how much oxygen, how much gas will get dissolved in the liquid, right? So we cannot do those type of things in the fermentation process. So don't bring that heat transfer concept here. That remains in the heat transfer, sorry, in the mass transfer. Okay, so at higher cell concentration, the rate of oxygen consumption may exceed the rate of oxygen supply, leading to oxygen limitation. So if the cell concentration is higher than what oxygen demand is, if the oxygen demand is higher than the supply, then oxygen becomes a limiting factor. The specific growth rate in that case varies with respect to the DO concentration to the saturation kinetics. And below the critical concentration, the growth respirator follows first order kinetics with respect to the DO concentration, and above the critical value, the growth rate is independent of the DO concentration. What it means is, what is the zero order reaction and the first order reaction? Which one is substrate dependent and which one is not substrate dependent? First order is substrate dependent, zero order is not dependent, independent of it. Can you see the relationship? If your concentration impacts if the DO concentration or dissolved oxygen is impacting your cell concentration, it's a first order reaction, right? So below the critical concentration, the growth respirator follows first order kinetics. Above the critical concentration, that you have enough oxygen, the cells will happily grow. So oxygen transfer from gas bubbles to cells is limited by oxygen transfer through liquid film surrounding the gas bubbles. When we get into the heat and mass transfer, you will see how the oxygen is transported. So let's say if this is your cell mass, like they might be in a clump, right? So your oxygen is coming out from your oxygen, like the sparger gives the bubble will give out the oxygen. So there is a thin layer that it has to cross, then there is a medium through which it has to pass, and then there is a layer over a cell mass where it has to pass, and only then it can be consumed by the cell, right? So it becomes like you have mass transfer through multiple mediums with different diffusivities. So the diffusivity here is different, the diffusivity here is different, the diffusivity here would be different. It's like a series resistances which will get accumulated, so it changes. Now if the sparger was working properly, there would be gas bubbles around the cell biomass, so the transport might be direct, so that will improve the overall oxygen transport. So dCl by dt, the concentration with respect to change in the time, is your KLa C star by Cl. This is your oxygen transfer rate, and then you have your oxygen uptake rate, so which is your QO2x is equal to x divided by y, x by O2. This is your yield with respect to O2. So when oxygen transfer is your rate limiting, your OTR is equal to your OUR, which means oxygen transfer rate is equal to your oxygen uptake rate. And this is the equation that you will get at the this is the equation that you get at the very end. So the growth rate varies linearly with the DO concentration under oxygen limitation. These are just the conceptual parts that you should be aware of. Another would be your pH. So redox potential affects the extent of oxidative reductive reactions. If the function of the dissolved oxygen, the pH, and the other is other ion concentration. So it's a function of that. So you are changing, if you change the pH, if you change the dissolved oxygen, if you change the ion concentration, you are going to change the redox potential of your system, and that will create changes to the oxidative reductive reactions. So, if your CO2 gets added to your fermentation system, CO2 can be toxic. So, that will impact your cell growth. The presence of ions affects the transport of nutrients across cell, and it also impacts the metabolic function and the solubility of dissolved oxygen. We are going to take a break now for 15 minutes, and then we will get back. What do you think, or do you want me to continue? I can finish it off. You want me to continue? Sure? Okay. So, let us solve this particular question if you want to. So, what you have here is your single cell protein is produced from methanol using a 1000 meter cube bioreactor. The biomass yield from substrate is 0.42 grams per gram. Biomass yield from substrate is 0.42 grams per gram. The Ks is 0.7 milligram per liter. Your mu max is 0.45 per hour. That means you are given with your mu max. The medium contains 4% weight by volume methanol, and a substrate conversion of 98% is desirable in the process. Now, methanol is being used to produce single cell protein. The medium contains 4% methanol weight by volume. Okay? Understand this difference, this ratio. So, for batch mode of operation, the initial cell concentration is 0.1 gram per liter, that is your X naught, and the downtime between the batches is 5 hours. Your T D is given. You need to calculate the annual biomass production. You also need to calculate if the desired annual biomass production is to be 10,000 tons per year, what should be the volume of the reactor. Okay. Now, let us say, so what is the total mass of cell produced during the batch reactor or the batch culture? X is X f minus X naught into V. This is your total mass of cell produced during the batch culture. And the total time for the culture is your T total. If you remember, we did discuss T batch time plus T downtime, TB plus TD. How many number of batches are they doing in a given year? So, the number of batches would be 365 into how many days are there? 365 days. How many hours are there? 24. If you divide it by the total time, you will be able to get the number of batches that are there. So, the number of batches carried out in 1 year is 365 into 24 divided by TT, and the annual biomass production which is your tons per year, would be X into number of batches. Clear till now? Now, out of all of these, what are the parameters that you have and what are the parameters that you do not have are the equations that we are going to look for, and we are going to choose and we are going to solve. If you go back a couple of slides, let's say, sorry, if we go back a couple of slides, no, we do not need to go back a couple of slides for this. So, what you have here, the very first thing that let us start with what your mu is. So, mu is your mu max S over S plus K s, right, and your mu will become mu max under a specific scenario when your X s is very less than your substrate. So, in this particular scenario, what is your K s, which is your 0.7 milligram per liter. Your S is pretty huge value as compared to your K s. So, in this particular case, we can assume that to happen. So, your Y x by S would be x minus x naught divided by S naught minus S. Your x becomes Y x by S, S naught minus S plus x naught. I have to keep the question open here. Side. Okay. Now, if you remember the equation where x was x naught e to the power mu t, so if I do ln x by x naught is mu max t b. This is your batch time. The t that we had here was your batch time. So, I am rewriting that equation, going backward into that. Okay. So, your T b will become 1 by mu max ln x by x naught, right. Now, x is x naught, sorry, x is y x by s, s naught minus s plus x naught. This is the equation that we have identified, right. So, your T b will become 1 by mu max ln 1 plus y x by s x naught s naught minus s f. So, this is how we will estimate the value of S, sorry, T b. Now, what are the parameters, what are the values that are provided to us in this particular question? So, if you remember, if we go back to the question, what would be your S naught or S initial? 4% weight by volume methanol and the substrate conversion is 98% is desirable, right. So, 4% weight by volume. So, your SI or S naught is 4% weight by volume. So, let us say if it is, that means 4 grams per 100 ml, right, that then only you will get weight by volume, right, in percentage, 4 grams per 100 ml. So, that means 40 grams per liter. Okay. So, now you have your S naught. What is your SF, S final? 98%, conversion of 98% is desirable in the process. So, 0.02, 2% is still left. So, your final concentration that would be left, 98% has been converted, 2% is left. So, 0.02 into 40 grams per liter will give you 0.8 grams per liter, right. So, now you have your S naught, you have your SF, I have this, I have this. Do I have Y x by S already? The biomass yield from substrate is 0.42 gram per gram, Y x by S is 0.42 gram per gram. Is given. You are given with your mu max. So, I have my mu max, I have my Y x by S. Do I have my X naught? Initial cell concentration is 0.1 gram per liter. I have my X naught. So, once you put in all of those values, you will get your TB to be 11.35 hours. So, your X final would be X naught e to the power mu max TB. So, your X final would be 16.5 grams per liter. This is your final cell concentration per liter. Excuse me. Clear till now? So, very first slide we had that the total mass of cell produced during the cell culture was XF minus X naught into V. Your XF is 16.5, X naught is 0.1, that means 16.5 minus 0.1, that becomes 16.4 into V. What is your V? 1000. So, your total cell mass would be 16,400 kgs, right. 16.5 minus 0.1 is already given. We have estimated our XF. What is the downtime? Is 5 hours, right. So, your total time is 5 plus 11.35, that becomes 16.35 hours, right. So, now 365 into 24 divided by 16.35 will give me 536 batches, and I know my X, I know my number of batches, that will give me around 8,790.4 tons per year. This is how we use these equations. Sorry for the mess. I just didn't want it to lose the slide of the question, because we have to come back to it again and again. So that's why I'm using all the space provided. Got the idea? Now, what was the last question? So, if the desired annual biomass production is to be 10,000 tons per year, what should be the volume of the reactor? Now, if 1000 gives you 8790.4 tons per year, right? How much will give you 10,000? That's rearrangement. So, your answer that you should get is 1137.6 meter cube. So, this should be the volume that should give you 10,000 tons per year. Simple. I had given this extra spaces to solve. This one is something that might relate to what you are going to do. Would you like to try solving this by yourself? It's pretty straightforward. So, a strain of Escherichia coli or E. coli has been genetically engineered to produce human protein. A batch culture is started by inoculating 12 grams of cells into 100 liter fermenter. So, your volume is 100 liters, 12 grams of cells is your inoculation, that is your X naught, and the fermenter containing 10 grams per liter glucose. So, the nutrient content is 10 grams per liter. The maximum specific growth rate of the culture is 0.9 per hour. Assuming that the culture is growing at the maximum specific growth rate in the exponential phase, it remains constant, mu is equal to mu max. The biomass yield from glucose is 0.575 grams per gram. So, you are given with y x by s, you are given with your mu max, x naught, s, anything else that I missed, the v, you have pretty much all the parameters. So, estimate the time required to reach stationary phase. Your x naught is 12 grams per 100 liters, which is your 0.12 grams per liter, right. We did that equation in the previous question, was Tb is 1 by mu max ln 1 plus y x by s, x naught, S naught minus S f. This was your equation that we have used for the batch time. Are you provided with your S naught? You are provided with your S naught, right? We are going to assume all the substrate was consumed. So, you will find your Tb in this case to be 4.3 hours. The other aspect is what will be the final cell density if the fermentation is stopped after only 70% of the substrate is consumed. So, your xf, so if only the 70% is consumed, your batch time will change. If only 70% was consumed, your batch time will change. So, you have to estimate your batch time again, and when you will do that, you will get, because your SF is now 0.3 of S naught, your S naught was 12 grams, 12 grams? No, 10 grams. So, it becomes 3 grams per liter, right. So, SF becomes 3 grams per liter. At that time, your Tb should come around 3.94 hours. So, because we changed the substrate, how much substrate was consumed, my batch time changed. So, your XF is x naught e to the power mu max Tb. Your final concentration of xf would be 4.16 grams per liter. Excuse me. C'est bon? Sorry, oh, you're still writing. You don't have to write these. These particular questions are available in your Schuler and Kargi if you have solved that in the past. See, the reason I gave you this particular question in the previous one, the intent is for you to understand what TB is, how we estimate those TB, how this equation that x is equal to x is equal to x naught e to the power minus ut comes into picture, what your mu is, what your mu max is. Now, what we are left with is the fed batch cultivation. We can start the fed batch cultivation after taking a 10 minutes break. Will that be okay? Okay, welcome back. I hope the break was long enough or not long enough for many other people who are still missing here. Okay, so we have discussed batch design of the batch reactor in detail. So, now we are going to get into the fed batch. So, when we started our conversation today in the morning, we were looking at 3 different modes of cultivation. One was the batch cultivation, fed batch cultivation, and then third was the CSTR or the continuous stirred tank reactor. Now, fed batch just like is an opposite, I will not say it's an opposite. So, in a batch reactor you have no input output. In fed batch at least you have an input. So, that is the only difference between the batch and the fed batch, it has an input. Now, what it does for us is that it gives us certain control. You will see in the further slides, but I will discuss a little bit about those type of controls. So, let us say if I want to, if too much of substrate is injurious to the health of my cells, I can limit the flow of my substrate, right. So, it provides me some form of control. If I have to have multiple types of substrate to produce for my cell growth, I can do that, or if I can regulate the growth factors that are required, I can do that. So, that is why the fed batch has a very versatile application in terms of where you can apply, and that is where we were discussing, like in some of the recombinant DNA applications that we have discussed in as part of the synthetic biology, you might be able to go back into those references and see that they have used fed batch as the growth process or the cultivation process of choice. So, fed batch culture is a semi-batch operation. So, nutrients are fed either intermittently or continuously during the course of the operation. So, if you have, let us say, this is a reactor, you have an input coming in, there is a limit to which we go in terms of adding the substrate concentration or the substrate of the total biomass would be there. So, the volume is at least up to 80%, not more than that. We still want to keep a little bit of head space. Any idea why we keep that head space? For gas mixing, like, or for the transfer. So, nutrients, the culture bath is harvested only at the end of the operational period once we have reached the full volume, either fully or partially. So, once it has reached the volume, I can remove the cell cultures and then I can restart. So, repeated fed batch till the cells remain fully viable and productive. So, we can keep on repeating it. So, one of the more feed streams, but no effluent. We can have more than one feed stream. So, as I said, if more than one carbon sources are required or if more than one nutrient is required, I can have that. And if the nutrients are my rate limiting, I can change the flow rate, I can control them. So, fed batch at least provides me some form of control. So, if you, in your work, if you require control for your product formation, fed batch would be a very good option. The culture volume increases continuously because you keep on adding the nutrients, right. So, the culture volume is increasing, you are not exiting out anything, you keep on adding. So, a fed batch culture is a dynamic operation. The concentration of the limiting nutrients in the culture can be manipulated, and the nutrient concentration profile can remain at a constant level or to follow a predetermined profile. So, either you keep it constant. So, let us say if this is your reactor, you keep the nutrient concentration or the flow rate constant, or you have put it as such that first half an hour this would be the flow rate, the second half an hour there would be another type of flow rate, like the flow rate will increase or decrease depending upon what you are trying to do. Sometimes if you give less substrate to your host or to the microbes, they might be developing a different type of metabolites, right, because metabolic pathways will change, they will try to adjust themselves. A batch mode is used to end the fermentations once it has reached that 80% volume, close everything, you run it as a batch. That is what it is used, it ends as a batch. So, manipulation of one or more feed rates, a mean of regulating the nutrient concentration, controlling the key reaction rates, what compounds should be fed and how they should be fed or how they should be added, those are the controls that we have. This benefits when your, let us say if there is a cell, as I was explaining at that time, if there is a cell that can actually use 2 or 3 different types of carbon sources, and depending upon the type of carbon source, it can develop different types of products, and your intention is to get all those products. So, let us say if I, help me out here, if my intention is to generate natural color with a different product also, like both of them have to be done at the same time with the host, whichever host I am choosing. For the natural color, the host prefers a specific carbon source, or if I have the carbon source, if it is less in concentration, it tends to go and produce only the product A. If I have more of my substrate, it produces product A, but it also produces my natural color. So, if my intent is to get more of my natural color, what will I do? I will keep on I will keep my flow rate high, right. I will provide it more nutrient. If it was other way around, I will keep my nutrient flow rate low, so that only my natural color is produced more. So, those type of controls are possible in a fed batch reactor. It is just an example. Okay. So, you can maximize the cell formation rate for a constant cell mass yield. The substrate concentration maintained at a value that maximizes the specific growth rate. Now, the mu max is the max at the concentration at which we can reach. If I can keep on changing, I will keep on maintaining my mu max, or I will keep on increasing it. It will always be in increasing order once it has not reached the plateau. The feed rate regulated to hold the substrate concentration constant at the desired value until the reactor is full. Once the fermenter is full, it is run as a batch mode. So, advantages of the fed batch: intermittent feeding of substrate and maintains low substrate concentration that can help in substrate inhibition. If my substrate is the inhibiting factor, I will maintain a lower substrate concentration. So, no inhibition. I can get higher cell density in fed batch as compared to batch. There is this catabolite repression. Example would be a penicillin fermentation process. If you are changing the concentration of your substrate, it might lead in the penicillin fermentation. The presence of the glucose basically leads to catabolite repression, and the same thing happens with respect to the baker yeast fermentation. If too much glucose is available, ethanol generation can take place because of the glucose phosphorylation. So, that is considered as a Crafty effect. So, if you are able to control your flow of your substrate in the fermenter, you are going to avoid these two types of issues. So, if you are going for a penicillin fermentation, what should be the reactor that you are going to choose, or the cultivation mode that you are going to choose? Fed batch, because that gives you the control, right, to avoid catabolite repression. If you are going for baker's yeast fermentation, fed batch. Auxotropic mutants or expressions of control genes, inducers and repressors. So, let us say if you want to have that mutated versions of no, not mutated, if there are genes, selective genes that needs to be activated, or metabolic pathways that needs to be activated in the E. coli or any of the microbes that you are putting into the fed batch, by changing the repressors or the promoters that you are going to send through as part of your nutrient, you can change the expression. So, extension of the operating time, supplement of water lost by evaporation, and decreasing viscosity of the cultured broth. Now comes the whole design equation, and this would be the main one. So, for the fed batch, now the whole equation will change. If you remember, we had in minus out plus generation minus consumption is equal to accumulation, right. So, in this case again your dx by dtv is your accumulation term. You have an in this time, let us say. So, there is an in for the feed and there is an in for the substrate. So, if I am only looking at the cell mass concentration, I am looking at f x naught. So, that is my feed in. Anything out? Nothing out. Definitely there is a generation, which is your V mu net x. This is a simple mass balance, okay. This is a very simple mass balance. So, since dx by dt can be written as, because the volume is not constant in this case. In the case of the batch reactor, the volume was constant. In the case of fed batch, the volume is not constant, it is changing by the time you are adding your substrate. So, that is why you will be differentiating that also, and that is the reason why you have this V dx by dt and x dV by dt, because you are changing the volume with respect to time. With respect to time. Then your Fx naught plus V mu x x is equal to V dx by dt plus x dV by dt. In the end, the equation, sorry, I will keep on repeating. This is the equation that is the most important. Your d is F over V. Now, your d here is dilution rate. F is your feed, V is your total volume. Mu is F over V. So, F and V is V naught plus F t, the same equation here, and the equation for the mu net is given as the Monod's growth model, a different model of that application. What is steady state? When things don't change with respect to time, right? What is quasi steady state?
When t tends to 0.
When t tends to 0?
I mean, del t tends to 0.
Explain it in a layman way to me.
It's instantaneous.
Instantaneous.
Yeah.
When the change is very, very small. So, let us say in a fed batch, your cell growth will continue till the time you have your substrate, right? The time it is about to finish, you start the tap, you add the substrate. So, your cell mass again starts growing up. Is it in a quasi steady state? It is in a quasi steady state scenario, but that is not the reasoning of it to be in a quasi steady state scenario. Your mu max is pretty much equal to your dilution rate. That is what happens in a quasi steady state. In this case, it will be almost equal to your dilution rate. So, mu net is equal to d, right? This is what happens in a quasi steady state. That is the reason I asked. Now, the total biomass, the final equation that we get in terms of, if you remember, we had that x is equal to x naught e to the power minus mu t in case of the batch. Same equation here would be x t is x t naught or y x by s s f into t. So, balance on rate limiting substrate, the ds by dt is f s naught mu x t by y x by s at quasi steady state. Essentially, all the substrate is consumed. So, no significant level of substrate can accumulate. Hence, as quasi steady state is reaches, all the substrate is consumed, more substrates comes in, no accumulation, then the new thing starts. So, no significant level of substrate can accumulate, hence f s naught is equal to mu x t by y x by s. This is an important parameter here. The most important equation would be this and this. the product formation in terms of the fed batch reactor, the final equation that if you do the mass balance for the product, the final equation that you will end up with would be the P is equal to P 0 naught V naught divided by V naught F T Qx QPx. Now, QP you remember, right, 1 by x dP by dt, the specific product growth rate. Let us solve this question very quickly. It is a very simple one. So, what you have is in a fed batch culture operating with intermittent addition of glucose solution, values of the following parameters are given at time t is equal to 2 hours. So, your time is given as 2 hours. So, when the system is at quasi steady state, so at quasi steady state, so at quasi steady state, so V is given as 1000 ml, S naught is given to you, the Ks value is given to you, X naught at time t is also given to you as X naught, S naught is given, F is given dV by dt, which is 200 ml per hour, mu m, which is your mu max, is given as 0.3 per hour, Y of m x over s is given as 0.5 grams dry weight cells per gram glucose. You need to determine V naught. Now, which equation will you use for the V naught? Do you see any equations here? Your V is V naught plus F t, right. So, your V is V naught plus F t. Your V naught is already given as 600 ml. You can estimate, right? V is given, F is given, T is given. You can estimate your V naught to be 600 ml, and your D, the dilution is F over V, which will come around to be 0.2 per hour. Now, for your second part, your S is Ks D by mu m minus D. Why? Where am I getting this equation from? S is given as how much? What is your Ks? Ks is given to you 0.1, D is already known, mu m is known, your S will come out to be 2 grams per liter. So, you have your S, you have your D. How do you estimate your X t? See, it is asking you to find S, X, X t and P at quasi steady state if Q p is given as 0.2 grams at a product per gram and the cells per hour is P naught is 0. There is no product at time t is equal to 0. So, I have found my S, I have found my, I need to find my X t. Now, X t from the equation would be X t naught plus y x by s, s naught f t. I am given by y x by s. So, x t naught plus f naught y x by s, s naught t. This will give me a value of 50 grams. Similarly, I have found my x t, x s, the p. I will be using the equation here, this one. The moment I put all the values, because I do have the f value, I do have the f naught value, I have the q p value, I have the x m value. Do I have the x m value? This is how you estimate the XM value. So, your P with equation would come around to be 16 grams per liter. You don't have to understand how we solved this, what this question was. You do need to understand how we solved it by just understanding what were the equations we used. But the most important part that you should understand from this whole lecture today is what is the main difference between the batch and the fed batch. What is the importance of the fed batch? What are the benefits that you get out of fed batch? And what are the benefits and advantages you get out of batch? Because depending on that, plus depending upon what you are trying to grow, you would be able to design your own process once you get into your case studies. Okay? We'll end the lecture today here, and we'll meet tomorrow again for heat and mass transfer and the scale up. Those are another set of equations and equations and equations. See you all tomorrow. Thank you. How do you find the XM?